Well, hello. This is Miss Dakenford again, and I wanted to share with you the start of Chapter 11, which would be getting into the process of natural selection and what some might call microevolution. That term has gotten out of favor for now, but so let's take a look at this. Let me go ahead and present. And we're going to do a little bit of math in this video, not too much. So in Chapter 11, it talks about the variations in populations and what this then can cause with the population. Now, we ended the other video just talking about what Darwin and Wallace had presented, and that would be the process of natural selection. We're going to talk about that a little bit more today. So first and foremost, let's take a look at the variation in populations. Now, we would refer to this as genetic diversity, and more genetic diversity is good in a population. So you take a look at the variety of butterflies, the variety of different color flowers in a population. More genetic diversity is going to mean that there's a wider variation in phenotypes, in the characteristics in that population. And that means that you're going to have a better likelihood that some in that population are going to survive. And that's always a good thing. So now all the genes in the population would be considered the gene pool. So if you were to take a look at all of the potential frogs that are out there, and the number of spots on the frogs and the size of the frogs, their vocal calls and their webbing, so all of those genes that dictate all of those traits, we would consider that the gene pool. So a bigger gene pool would represent more genetic diversity, and we like that. In fact, endangered species, one of the scary things about endangered species is when there's so little genetic diversity in the population that even if we were to save them and have them reproduce, we worry about their lack of genetic diversity, which is why zoos that have captive breeding programs, they try very, very hard to expand their breeding programs to zoos and different zoos depending on when they breed their animals. So genetic variation comes from mutations, and we looked at mutations before Christmas. Mutations can be good, neutral, or harmful, and of course that somewhat depends upon the environment. Genetic variation can also come from the gamete production, the different varieties of gametes that an animal or plant can make, that random assortment, recombination that occurs when the chromosomes cross over, and then which sperm cell fertilizes which eggs. So all of these can contribute to the variations of phenotypes in a population. So what does this then mean for our populations? So let's take a look at this and remind ourselves what an allele is and what we might refer to as allele frequencies, because that's going to help us measure what's going on. So as we're talking about natural selection and evolution, let's get a hold on how we can actually measure this, and that's going to be our goal. So allele frequencies are a measure of the variation in a population. So let's take the example of the frogs. So how many green frog alleles are out there versus brown frog alleles? So one thing to remember is that each frog has two alleles. You get one from mom, one from dad. Hopefully that's not a huge thing to remember. We just did this a few weeks ago. So notice if I have my brown frog over here, let me get my pointer here. If I have my brown frog, and let's say, for the sake of argument, let's say the brown frog is recessive, And so in order to get a brown frog, we have little g, little g, little g, little g, sorry. And then for the green frog, let's make this green frog a heterozygous, and then this green frog a homozygous dominant. Now, this gives us a way to measure the alleles in the population. So for instance, let's think about this. If there were 10 frogs that were homozygous green, then there are actually 20 green alleles. Remember, each frog has 2 alleles. So you would then say, well, I've got 10 frogs, they're homozygous green, so that would be 20 green alleles out of 20 alleles. So the frequency would be 1. Or you can also express this as a percentage, 100%. But what if there are only 8 frogs that were homozygous green? Then we would have 16 green alleles there. What if the remaining 2 frogs were heterozygous? Then, because they're heterozygous, we have 1 green allele in each frog. That would be 2. And so if I were to ask you the frequency of the green allele in those 10 frogs, you would say 16 plus 2, that would be 18, divided by 20. And so now we have a way of measuring if we knew which frogs are homozygous and heterozygous. Now, we're not going to do too much with this math. This is really an introduction to this. So we're going to do this more as a conceptual problem. But it gives us a way to now sink our teeth into this problem of how do we measure whether or not a population is changing. So let's take a look at this and at least get some practice with this. So let's see, we've got 4 brown frogs, and then I have 6 green frogs. What I'd like you to do is think about how we could measure this. What's the frequency of the brown frog allele, and what's the frequency of the green frog allele? Take some moments in class and see if you can figure this out. So hit pause and discuss. So hopefully you were able to think through that, and your teacher has the answer key for that, and you guys can discuss it. So here's another example. This one's not a little bit of not as clean. It's not out of 10. There are 12 total alleles for skin color traits in this gene pool. And so you can see how they figured it out. There are 6 frogs. That's how they got 12. And so they counted up the number of green. 1, 2, 3, 4, 5, 6, 7. So you can see where they got the 7. 7 out of 12. And then we have frequency of the G allele 5 out of 12. And so notice they should add up to 100% or 1. And now, that gives us a way to see whether or not the population changes. Now, a couple of researchers a number of years ago created this equation, and they're called Hardy-Weinberg. Well, one was Hardy, one was Weinberg, and they came up with a system by which we can actually have a metric. And so the Hardy-Weinberg equilibrium gives us a standard to base our population. And that allows us to then know if the population is changing. So what I want to leave you with is this idea that if the population is not changing, then the allele frequencies are going to stay the same. Now, that doesn't mean the population doesn't, like frogs can't be eaten. I mean, obviously frogs are going to be eaten, frogs are going to be born, frogs are going to die. But if it's a stable population, then we should see the same frequencies exist in that population. If the population is changing, then we should see the frequencies change. And I'll give you an example to let you think about it. In fact, let me go back. What if, because these are variations, remember some variations are adapted better. Let's think about our typical pond, and let's say this pond has a lot of green vegetation, and so the green frogs can hide better. What do you think would happen to the population of the frogs in terms of the number of green frogs and the number of brown frogs? What if the green frogs can hide better in the green vegetation? What do you suppose is going to happen over the next few breeding periods of the frogs if the green frogs are going to do better, and then the brown frogs can be spotted by the predators? So think about that, and that'll give us something to end our video and start a discussion.